Today our first speaker is Jill Smith. She got her medical degree at the University of Florida, and I'm sure she's happy she's not in Gainesville, Florida today after Hurricane Irma. And then she got a residency at the University of Missouri. She then became a professor of medicine at Penn State University. For a few years, she came here to NIDDK and was in charge of the translational research. And now she's back at Georgetown University. She's a professor of medicine, and she's going to talk to us today about translational research, bench to bedside, clinical trials. Jill.
Hello? Oh, it is on. Okay. Can we leave that open? We're leaving that open because there's some fragrance in here that they sprayed that I'm allergic to. So, well, thank you, Dr. Moody. Thanks for having me. So, of course, I have some disclosures that I need to reveal here. I'm a co-inventor. It's now on 11 patents related to pancreatic cancer, and I do some consulting. I don't have anything to clip it to. But then it has to be this way because I look at the.
Do you mind taking it?
Can I take it off? Okay. Okay. Okay, I'm a consultant to some companies. So today we're going to talk about understanding how an idea is taken from the research lab to patient care. We're going to learn about the steps of conducting clinical trials, and we're going to try to comprehend the obstacles there are to overcome drug development. And I'm going to give you some examples of my translational projects. And can you hear me over that machine out there? Okay. So how many of you in here are graduate students or working on your PhD? Nobody. How many of you are MDs? No? Okay, okay. And everybody else is? Huh? Postdocs? So you okay, postbacks, okay. Okay, good. Desire to do one or the other, I hope? Okay, all right. So one of the things about research development is there's a lot of drugs that are tested preclinically. And unfortunately, the majority of those do not make it to the clinic. And so we have this bottleneck of development with drug development. And so that's a problem. So, or is the bottleneck really Washington? And I say that this is not really the bypass, but you know, part of the problems are the politics of doing research and the funding problems and whatever. So in order to do a research project and actually to do translational research, you have to have a good idea. And it has to be hypothesis-driven research. And, you know, you have to look at what is the problem that's going on and what needs to be done to solve this issue, and what can your research do to help the problem. So I'm going to probably close this if I can. Yeah, it's a little loud. Okay. Okay. And the other thing is that you really have to have passion about what you're doing. If you don't like what you're doing, you got to do something else. You have to like your research. You have to be excited about your field. You have to be excited with every little discovery that you have in your data. And that's very important. So as far as the phases of clinical trials, it's phase 0 to phase 4 that we're going to be talking about today. And phase 0 is where most people are working in the bench. And you have to identify an agent and is it hitting its target? So did you discover something that might be a good target for cancer therapy? And then you test it, of course, in cancer cells, you test it in animals in vivo. And then when you have done that, then you can take it to the next phase, or phase 1. And that's how you find out what the drug-related toxicity, or the DLT, or the maximum tolerated doses that you could treat in humans. And then the next phase is phase 2, which you're really looking at the feasibility and seeing, is it really better than a placebo? Then you have phase 3, which you look at clinical outcomes compared to control arm. And then phase 4 is really post-marketing once it's already approved. So these are also some of the different types of clinical trials. And there can be treatment trials with drugs, there can be prevention trials, early detection screening trials, diagnostic trials, quality of life trials, genetic trials. And the trials can either be industry-sponsored or investigator-initiated. And most of what I'll talk about today is investigator-initiated, because most of us are working in a research lab and you come up with an idea and you'd like to see your idea go all the way through to the clinic to help people. That's the whole reason why we're doing the research. So phase 1 usually only involves about 15 to 30 people. And the whole purpose of it is to determine if the dose is safe. Sometimes they do this in normal controls rather than in cancer patients. But obviously, if you're testing a new chemotherapeutic drug, you don't want to give that to normal controls. So you would test it in probably the cohort that you're looking at. For example, I'm going to talk about pancreatic cancer. And then you want to know how the treatment should be given. Do you have to give it intravenously? Can you give it by mouth? What are the pharmacokinetics? How long is the half-life? And, how do the treatments affect the body? Does it cause liver toxicity or other sorts of toxicity? And then phase 2 is typically, can be less than 100 people, and it has to have a primary endpoint. It's usually done in an unbiased or blinded way. And so you want to know, what is the efficacy? Does this new treatment actually work? And is it more effective than a placebo? And it doesn't really compare it to other treatments in a phase 2. You're just comparing it either sometimes to standard of care or to placebo. And then phase 3, it can be anywhere from 100 to thousands of patients. And there's an equal chance of being assigned to one of the 2 or more groups. And it determines how the new treatment compares to the current standard therapy. Or again, it can compare to placebo. And in this category, there can be superiority and non-inferiority trials. So a non-inferiority trial is that you want to just show that your new treatment is just as good as the standard of care. So maybe it'll be approved because it's less expensive, or it's easier for patients to tolerate, but it's not any better than the standard of care. So a superiority is that it is better than the standard of care. And then the phase 4 trial is usually in hundreds to thousands of people. For example, the hepatitis B vaccination, when it came out, this was taken across the whole globe. And usually it looks at the drug's risks and benefits, and it's called post-marketing. So after it's already been approved and it's out there on the market. So one of the things we typically do when we're going into a trial is we have no idea what the sample size or how many patients you need to include. And so often you do what we call a pilot trial, and that may be just a small open label trial that you're going to treat a small cohort of patients with your study drug, and you want to see, if there's any effect. And if you see maybe there's a 30 or 50% response rate, then you will use that information to help you design a bigger and more pivotal trial. So that will give your statistician some basic information about how effective the drug might be when it goes against a placebo. So this will help you calculate the sample size as you move into phase 2. So when you're doing a randomized clinical trial, there's supposed to be an equal chance of being randomized to one or two of the treatment groups. And one group usually gets the most widely accepted treatment or standard of care, and the other gets the placebo. And it is important that they try to be balanced. So when you're randomizing your patients, you would like to make sure that if your outcome is going to be, say, quality of life, you want to make sure that when you randomize patients at the onset, that all the patients with the worst quality of life don't end up in one treatment arm versus the other. So you want to make sure that they're balanced. Most people balance for age, for gender, and maybe if there is some efficacy point that you're looking at, like response rate, like RECIST criteria on CAT scan, you want to make sure that everybody who is in treatment A is not stage 3 cancer and everybody in treatment B is stage 4. So they should be balanced at the onset. Whenever you're doing clinical trials, it's very important that patients' rights are protected, and there are ethical and legal codes that govern the medical practice in clinical trials. All patients have to sign and understand an informed consent. There are review boards. Most institutions will have their institutional review board, and there are, before that, there's other scientific reviews. For example, if it goes through a review when you're submitting a protocol to be funded by the NIH, there's a peer review and scientific review that reviews that protocol. There are also at other medical centers, like at Georgetown, we have Lombardi Cancer Center, and we have our own cancer scientific review. And they will review it and make sure everything is ethical before it moves on to the IRB. And then after that, there's also a data and safety monitoring boards, so that when the study is going, you want to make sure that somebody is monitoring, how it's doing, and even especially if it's being done in a blinded fashion, because if everybody who's getting the treatment is living much longer and everyone on the placebo is dying, they may unblind it and end up treating everyone. So there may be early stopping rules that your data safety monitoring board will go by to determine if the drug is a benefit or if it's detrimental. If they find that everyone on the treatment arm is ending up with liver toxicity, they may terminate the study prematurely. And of course, most recently, if genetic testing is going to be done, that has to be added to the consent. And today we store a lot of blood samples, and people will go back to them later in time and do some GWAS studies or RNA-seq, and they're looking for some genetic alteration. If blood samples or tissue samples are going to be stored, there has to be included in the consent form a paragraph about the risks and benefits of that. Because if you discover that this patient has a hereditary, SNP that may worsen their long-term survival, and if insurance companies find out about that, they could lose insurance. So all those things need to be stated up front in the consent form so that patients are aware and they can elect to have their samples removed and not kept in storage with the new genetic codes. So how do you do it? What are the nuts and bolts? And I'll just give you some examples from my experience and some studies that I've done. First of all, it takes a whole research team, and that's very important. No man is an island anymore. There can be the principal investigator, there's co-investigators, but there are other very, very important key players that will help a study be successful. One of those is the nurse or the study coordinator, who will make sure that all the documents and the patients are taken care of properly. Having a statistician is extremely important because they tell you your sample size and then they'll crunch your data when you're done. I had one study that had over 100 patients in it, and I just handed the Excel file with the data in it over to the statistician, and they did all the analysis. And it's actually better that way because you don't want the person who's in charge of the study doing the analysis. That way it is, not biased to one group versus the other. And the statisticians usually don't know which is the treatment group. They just know group A and group B. So that makes it a much more rigid study. You should have a data manager. And whenever they're entering the data, The data manager may not know what the normal range is for, say, an ALT and the liver enzymes. So usually we have a place in our Excel files if they're entering something and it falls outside the range, or they're entering a wrong number by mistake, it'll pop up red. So there are a lot of ways that you can foolproof this so that they don't enter some data in error. And then, of course, we talked about the Data Safety Monitoring Board. And you have to have an investigational pharmacist, because when this is done in a blinded fashion, the only one who really knows what the patient's getting is the pharmacist. And if there's a problem and you have to unblind the study, it's the pharmacist who knows what they're taking. So before you start the trial, there's several things you need to do. First of all, you can't do a clinical trial with a drug or a device in humans without approval from the Food and Drug Administration. And so you have to apply for an IND or an investigational new drug number. And I show you an example, the 1571 form that I have here. Then you also have to write a protocol. And that's the study design and what you plan to do and what the outcomes are. You have to have a consent form that the patients will be signing, and that has to be approved. You have to get approval by the Institutional Review Board. And then the hard part is finding a sponsor, getting funding and getting money to do your study. And then there's a lot of responsibilities of the principal investigator. Overall, the principal investigator is responsible for everything. Everything from submitting the information to the FDA, to the IRB, to overseeing the nurse coordinators and the patient care. And then you have to register everything on the clinicaltrials.gov website. This is required if you want to publish your results. And it has to be registered before patient number 1 signs the consent form. If the patient signs the consent form and you have not yet completed and finalized the registration, you are not allowed to publish your data. So keep that in mind. So this is an example of a 1571 form, one of the ones that I filled out. And you have to put in this one, every time you send it in, you have to put your IND number in the little box, and then there's another little box for the serial number. So the first time you submit your application and your protocol and you're getting approval, it's serial number 0000. And then the next one that you send in, even if you're sending a report saying, I'm changing the protocol to this paragraph to the next paragraph, you have to submit the changes, and you put in here, you know, what you're submitting in the report down at the bottom, and then the next serial number will be 001 and then 002, etc. So you have to send them in sequence. And this, it's all now done electronically through the FDA, and you have to do this for every time you submit something to the FDA and communicate with them. The other thing that's really important with your research before you take it to a clinical trial, or even before you go out and present a poster at a meeting, you have to make sure that you've secured intellectual property. So you may not have the patent yet, but if you have filed a provisional patent, which is very inexpensive, it's only a couple $100, and the institutions will do that for you, it protects you. And so you should do that before you go out and present anything publicly, even at a poster session. So the other thing is that the patent belongs to whoever you're working for. So for me, you know, if I'm working for Georgetown and I have a great discovery, they're the owners of the patent because they pay my check. So, but that's important. And if for some reason the university or the NIH or whatever chooses not to pursue your patent, they don't think that it's going to go anywhere, the inventors have the right to get that invention back and file it themselves. And I did that with one of my patents, which is now licensed and is being approved by the FDA to treat Crohn's disease. So, so it's important that you stay on top of things. Finding a company, there's a lot of biotech companies out there, and They're probably easier to deal with than the big pharma, because big pharma likes their own ideas rather than dealing with universities. This is just my opinion. But then you want, when the patent comes out and it issues, you do want to license it to a company, because you can't do, once you're an investigator, you have to turn it over to a company so that they will develop it. They have the resources and they can do the multi-center trials. So that's important. But the very important thing about when you license your patent, you should have milestones in there for whatever company you're dealing with. And if the company does not reach their milestone and they don't develop your drug or your idea in the time frame that they're supposed to, there should be a clause that you get it back. Because there are companies that are out there that will take people's ideas and just set it on the shelf to kill the idea so that they can come up with a competitor. So that's very important. This is just one of the patents. This is what a patent looks like, and this is just one of my patents that issued in 2014. So the next thing you need to do is you need to write your protocol, and there's kind of an outline on how you have to do your protocol. Everything from the title page to the summary. Usually you do an overview summary of the whole project, and then that's followed by the 100-and-something page document of the entire protocol. And this is just one of my protocols. I'm just showing you some examples of things that we're working on. And you have to have, I don't know how this pointer is working. You put your protocol number there. You put the title of your protocol. If it has a number, you want to say what clinical stage it is. So this is a stage 1 and 2, how long it's going to be, what's the investigational drugs that you're using, what's your research hypothesis, and then you just spell out what's your primary, secondary objectives. What are you going to do by this study? So in this study, we're doing a phase 1, and we're going to be trying to find out what the recommended phase 2 dose is by doing the phase 1, and then we're going to move into the phase 2 part of the study. And then, of course, you want to say how many centers are included, how many patients you're going to treat, inclusion, exclusion criteria, et cetera, et cetera. So that's how you have to do, that's the outline for your protocol, and what your drugs are and what the dose is, just kind of a summary, and then you go into detail in the full protocol. So the aims of a phase 1 trial and the first in human trial is really just to look at the safety and toxicity, determine the maximum tolerated dose, look at any pharmacokinetics. And I did some work with a drug called OGF, opioid growth factor, and you want to study the route of administration. Well, when we did this study, we used the typical dose escalation study. It's called the classic 3x3 design, where you enroll 3 people at 1 dose. And the dose you choose should be, you start off about 100 to 1,000-fold lower than the dose that you used maybe in animals. So when I did this study the very first time, I knew that I had used a certain dose in my mice, and so we used the lower dose. And the very first patient I gave it to ended up in the hospital in the emergency room that weekend. And I had to make a decision, was it my drug that made her get sick, or was it because she had metastatic pancreatic cancer? And so I went ahead and I enrolled the next 2 patients, and they did just fine. And so we escalated. We actually ended up going from the 25 dose all the way up to 250. So the patient actually had just gotten sick because of her cancer. So I'm going to tell you a little bit about some of the research that I do with pancreatic cancer and some of the clinical trials we're working on, and how you can take an idea from the bench to the bedside. Is anybody here working on pancreatic cancer? Nobody. Okay, well, come on, I need some competition. Well, it's the 3rd leading cause of cancer-related deaths in the United States, about 53,000 people a year. And the median survival, unfortunately, is only 3 to 6 months, and only about 6% survive 5 years. This survival rate has not changed in the past 50 years. In spite of all of our technology, all of our PET scans, all of our diagnostics, all of our treatments, nothing has been effective. So the death rate from pancreatic cancer is actually increasing. And you can see over here that it is it's already surpassed colorectal cancer, and it's surpassing breast cancer, and it's going to be soon, can't get this to work, second to lung cancer by 2020. So these are our current treatments for breast cancer with chemotherapy, and the one on the left is FOLFIRINOX. It was compared to the standard of care gemcitabine, and they got a survival of about 11 months. But you can see as you get down the numbers towards the end, there are very few patients still alive. Those are the number of patients. And then our other standard treatment is gemabraxane, which is nab-paclitaxel compared to gemcitabine alone, and that increased survival from 6 months up to about 8 months. So neither one of those that's our standard. That's the best we're doing today. And neither one of them is even at 1 year. So why is the prognosis so poor with pancreatic cancer? Well, one of the problems is that 90% of the people who are diagnosed with pancreatic cancer, it's already spread beyond the pancreas. It's very metastatic, and because of that, they're not amenable to surgery. That we don't have any screening tests for patients who might be at high risk. We don't have, like breast cancer, you can screen with mammography. We can't do screening colonoscopies like we can for colorectal cancer. So there is no screening test, and the current CAT scans and MRIs are not sensitive enough to pick it up in the early stages. And it's also resistant to chemotherapy, and it's resistant to immunotherapy. Everything that we have tried to treat pancreatic cancer with has failed. So my research, and Dr. Moody's an expert in this field, is on CCK or cholecystokinin receptors. And there's 3 types of CCK receptors, easy to remember, A, B, and C. A was originally called A because it was discovered in the alimentary tract, and B was originally discovered in the brain, and C is a receptor that we found that's a splice variant that's only found in pancreatic cancer. So the CCK receptors are the classic G protein coupled receptors that have a 7-trans membrane domain, and the CCK and gastrin are the major ligands for the receptor, and they have the same terminal pentapeptide, and that's how they can bind to the same receptor with different affinity. So what is cholecystokinin? Just to refresh you with your physiology, this is a liver, that's a pancreas, that's a gallbladder. So whenever you eat, and especially whenever you eat something high in fat, free fatty acids and certain amino acids get into the second portion of the duodenum, and they stimulate the release of CCK from the eye cells in the duodenum. And CCK is a classic hormone, so it gets into the bloodstream and it circulates to the receptors in the pancreas, and it causes the pancreas to secrete digestive enzymes to help you digest your food. It also causes contraction of the gallbladder so that the bile will help you emulsify your fat and digest your fat. And it causes relaxation of the sphincter of Oddi so that the bile can come out and help you digest your food. Gastrin is the other peptide that binds to CCK receptors, and gastrin is made in the G cells of the antrum of the stomach. So the stomach is divided in half. You have the endocrine part of the stomach and the exocrine part of the stomach, and the G cells release gastrin. They get into the bloodstream, and they act on the enterochromaffin cells, which also secrete histamine. If any of you are familiar with Pepsid AC or Zantac, any of those things, those are histamine blockers, H2 blockers, and they block acid by blocking the histamine. And then we have the proton pump inhibitors, So regardless of whatever is stimulating the parietal cell to secrete acid, whether it be the histamine, acetylcholine, or gastrin, the end result is the proton pump, which secretes the acid. And so if you don't secrete, if you block this with a proton pump inhibitor like omeprazole, Prilosec, Prevacid, Nexium, the little purple pill that everybody says is so good for their heartburn, the problem is that it blocks the normal feedback loop. So if gastrin is being, if the proton pump inhibitors are blocked here, gastrin levels will go up because you're not making enough acid. And when that goes up, one of the things that I see when I scope these patients is you get all these polyps in the stomach. And these polyps are because the enterochromaffin cells are stimulated, and gastrin is a trophic hormone, and it stimulates growth. So it stimulates growth, and in animal models, it can cause carcinoids and gastric cancer. So we found that the CCKB receptors are actually overexpressed in pancreatic cancer compared to normal cancer. And we found that if you treated cancer cells with gastrin or CCK, that it stimulates the growth in cell culture. And that's on the far right. And what we found when we looked at what type of receptor was on the cancer cells, we found that if we treated the cancer cells with gastrin, we could stimulate the growth. But if we added it with a CCK antagonist, it did nothing. But we could block the effect with a CCKB receptor antagonist. So that let us know that even if the cancer cells have both A and B receptors, it's only the B receptor that's responsible for the growth. And then we showed also in animal models, if you treat the animals with gastrin, you can stimulate the growth of their pancreatic cancer, and you can block the effect with an antagonist to gastrin receptor or the B receptor. Well, we found that the gastrin and CCKB receptor are actually turned on early in these precursor lesions called Pannin lesions in the pancreas. And a Pannin lesion is the same thing to pancreatic cancer as a polyp is to colon cancer. It's a precancerous lesion. And they get turned on very early, and it causes it to progress. So we stained these, we had some transgenic KRAS mutant mice that spontaneously get these Pannin lesions, and we showed that if we stained it for CCKB receptors, that they were markedly overexpressed in the mouse pancreas, which is really surprising because the mouse pancreas usually has the A receptors, but under the influence of KRAS, they make B receptors. So we took our transgenic KRAS mice, and we put some in their drinking water, a CCK antagonist to block the receptor. And then after several months, we took the age-matched mice, and on the top, what you can see is a control mice, that their pancreas was completely replaced with these precancerous lesions and some early carcinoma. Whereas the mouse that was on the CCK receptor antagonist therapy at the same age had very few, if any, of these lesions. And we had waited actually till the mice were 3 or 4 months old till we started the treatment because we wanted to make sure that they all had some pannins. This is an islet cell here in the pancreas. So with pancreatic cancer, we talk about pannins, which are the precancerous lesions, but they actually, if you cut out the cancer, they only make up about 20% of the whole tumor. And the rest of the tumor is made up of stroma or fibrosis. And there's a lot of things that play a role in controlling the growth of pancreatic cancer, including that there's a lack of tumor infiltrating cells, and there's inhibitory T regulatory cells that suppress the normal immune system from fighting the pancreatic cancer. So there's a lack of the CD8 cells and an increase in the T regulatory cells. And then this fibrosis, it's thought that that may prevent our chemotherapy from getting into the cancer. And there's been studies that show that the more fibrosis that's there, the less people will respond to the chemotherapy. So this is a study done by Berna that showed that there are CCK receptors actually on the pancreatic stellate cells, and these are the fibroblasts that cause all that fibrosis of the microenvironment. And this was a picture. We stained them for the CCKB receptor, and they were positive. So we did a similar study in our mice, and we wanted to know, well, if we put them on the CCK antagonist, can we block the fibrosis too? And indeed we did. So the blue stain is a Mason's trichrome stain, and you can see that the age-matched mice, there's much less fibrosis because the CCK receptor antagonist blocked the fibrosis. And then the third cell that CCK receptors are found on are the immune cells. And the immune cells, meaning the lymphocytes, have both CCKA and CCKB receptors. And that's important because as the cancer progresses from normal to cancer on the far right there, the T cells, the T regulatory cells increase, and they suppress our own host immune system from fighting off the cancer, and the CD8 cells, or the T killer cells, decrease. So we did some studies, and a lot of research is changing. We used to do human cancer studies in nude mice, but the problem is that nude mice don't have T cells. And so if you're interested in looking at the immunity or what's going on in the microenvironment, you have to use a mouse with a normal immune system. And with all of our new immune checkpoint antibodies, you have to use an animal model that has an immune system. So they've been developing more and more animal models that are more similar to humans that have an intact immune system. So in our studies, we use C57 black 6 mice or our transgenic mice that have an intact immune system, so we could study immune checkpoint antibodies. So we discovered that one of the problems with pancreatic cancer is that it doesn't typically respond to immune therapy. And the reason it doesn't respond is because there's no CD8 cells in the tumor for it to attack the tumor. And then also you have this fibrosis and the T regulatory cells in the microenvironment that prevent it from the immune checkpoint antibodies from working. So we came up with a hypothesis that if we treated pancreatic cancer with CCK receptor blockade, we could decrease the fibrosis and we could alter the immune signature of the microenvironment and make it more susceptible to immune checkpoint antibodies. And actually that's what we found. So these are animals who were getting PBS or the immune checkpoints or the antagonist by itself, but when we added them together in the combination, the tumors did not grow. And the reason they didn't grow, we looked at the inside of the tumors immunohistochemically, and we found that the CD8 cells go up when we give a combination of the immune checkpoint antibodies along with our CCK antagonist, and the T regulatory cells go down. We also showed that this improved survival of our mice, and that only the mice getting the combination therapy were alive at the end of the study. The other important finding was that we saw that only the CCK antagonist reversed the fibrosis, not the immune checkpoint antibody. So the immune checkpoint antibodies by themselves were not effective. So what we've done now, since we completed that in animals, we've actually designed a clinical trial to do the phase 1, phase 2 trial. And we're going to use our CCK receptor antagonist along with an immune checkpoint antibody. And we're going to do a pilot trial in patients. We're going to figure out what the maximum tolerated dose is of our CCK antagonist. And we're going to work with a company that has already approved our protocol. And we're going to treat patients who have pancreatic cancer. So where we are right now, we secured intellectual property. We're getting the IND number. We have a source for our antagonist. I have an industry partner. And of course, I'm waiting for the funding, and hopefully that will come through. So now the other problem I mentioned with pancreatic cancer is that there's no biomarkers. There's no way that we can screen patients that might be at high risk. CAT scans, PET scans, MRIs are not sensitive enough. So what are we going to do? And are there any blood tests or anything that we can do to evaluate patients? So many years ago, when I was doing just a regular PCR, trying to figure out what type of receptors pancreatic cancer had, I found that compared to normal pancreas, there was this bigger PCR product in some of our pancreatic cancer patients. And you guys don't even do Southerns anymore, but that's a Southern blot below that. But there was about a 200 base pair difference between pancreatic cancer and the CCK receptors in normal pancreas. And actually what we found out was that in up to 35 to 40% of patients who had pancreatic cancer, they had a single nucleotide polymorphism that caused a missplicing of the CCKB receptor gene. And when this missplicing occurs, the fourth intron, which is about 200 base pairs, is not spliced out, and it's translated into protein. And so this correlates with a SNP that we found. And the reason why we couldn't do this in mice, and it never would have been found if we just were working with mice rather than human blood and tissue samples, is because in humans, the fourth intron has an open reading frame, but in mice and rats, there's a stop codon. So we could look all we wanted in our animal models, and we would never have found the splice variant. But we did find that there is this SNP. And the SNP is very important because it may predict who might be at risk of getting pancreatic cancer. Similar to the BRCA2 with breast cancer, BRCA1 with ovarian cancer. So the other important thing is that where this occurs is in the third intracellular loop. And what it does, that's the part of the CCK receptor that stimulates growth. And so we hypothesized, well, if they have this extra piece because the fourth intron is translated into protein, maybe people who have this SNP grow, their cancers grow much faster and they have accelerated growth. We did make an antibody to our SNP protein that distinguishes it from the regular CCKB receptor. And you can image the cancer cells that have this genotype for the SNP, and they express the spliced variant, or we call the CCKC receptor. So we did a pilot trial, and we took 110 patients, and we took blood samples from them, and we confirmed it. We also did tissue samples, and then we saw that their survival was definitely affected if they had the SNP or not. So then we did a larger trial on almost 1,000 patients to confirm our results. And so this is a validation trial, and that's another type of clinical trial that you can do once you do your pilot trial. And we did show that there was definitely a difference in survival in patients who had the SNP. They didn't live as long. So we're hoping that now that we've identified this SNP in pancreatic cancer, by easily doing a blood sample or doing a buccal mucosa swab, we might be able to identify people that are at high risk of getting pancreatic cancer. So the third problem is that chemotherapy is toxic, and it doesn't specifically target pancreatic cancer. A lot of cancers, if you can identify a receptor on the surface or on the cancer, whether it's HER2/neu or whether it's an estrogen receptor, then you can actually target your therapy. So there are no targets so far for pancreatic cancer, and there's also no imaging tests that have been effective in determining it early. So we decided we would develop a nanoparticle that would target the CCK receptor, and we call it a theranostic compound. So it's a therapeutic and it's a diagnostic compound. So what's a nanoparticle? And this kind of puts it in perspective. Nano means that it's somewhere between 10 and 100 nanometers in size. And just to summarize why we use the CCK receptor as a target If you look on the left, in the normal pancreas, there are some CCK receptors on the cell surface, and CCK or gastrin can bind to the receptor. And when they do that, they secrete your digestive enzymes. But in pancreatic cancer, cancer is not interested in digesting your food. The cancer is just interested in proliferating. So when the receptor is stimulated, the cancer cells grow. And then what happens is something turns on gastrin, and the gastrin starts being produced by the pancreatic cancer cells, so it stimulates its own growth by an autocrine mechanism. And then this little loop here is the splice variant CCKC receptor, which is present in about 40% of patients. So we've developed a couple different types of nanoparticles. One of them is a nanoliposome. One of them is a calcium phosphate nanoparticle. And we can design them so that they can carry siRNA, and it can knock down gastrin that's in the cancer. And we put the target on the outside so that it'll bind specifically to the CCKB receptor. And then we could also load these particles with either a fluorophore or magnetite so that we can identify it by imaging or with MRI. So we've also designed a special way that we can bind a DNA aptamer that we found by doing the CellX method. And this aptamer binds selectively to the CCKB receptor and it does not stimulate growth. So we have taken this aptamer and we've covalently bound it onto nanoparticles. And so the nanoparticles will deliver whatever we want them to directly to the cancer, and they'll bind to the cancer, to the CCK receptor. This is just a picture of a couple of mice here on the left. These mice were injected with nanoparticles that had indocyanine green in them for fluorescence, and they were not targeted to the CCK receptor. And at 7 hours and 24 hours, you can see that there's really nothing left. However, in the mouse that has an orthotopic pancreatic cancer growing in its pancreas, when they were injected with the nanoparticles, with the targeted nanoparticles carrying the fluorophore, you can see at 7 hours and still 24 hours later, the particles are still concentrated in the tumor, in the pancreas. So we've now designed a particle that will target selectively pancreatic cancer, and it can carry fluorophores. So we might be able to use this for early diagnosis and imaging in people who have smaller early cancers. Another one of the nanoparticles that we've designed is called a polyplex micelle, and it actually is made up with a polylysine tail, which is positively charged. And when we add it to our siRNA, it forms a micelle. And we have our aptamer on the outside here, so it'll bind to the receptor. The siRNA is on the inside, so it's protected from the blood and does not degrade. And we have polyethylene glycol, so it doesn't conglomerate in the blood, and the blood cells don't take it up. And we've already done studies in mice where we show that we can get this nanoparticle inside our cancer cells. We can actually, we delivered siRNA to gastrin to see if we could knock down gastrin, and we could knock it down by qRT-PCR, and we showed that the gastrin was also knocked down by peptide. And then when we treated our animals, only the animals that got the nanoparticles that were selective for the CCK receptor and carried the gastrin siRNA had decreased growth. So there's great potential for this platform that we could target whatever your receptor of choice is. It can carry whatever siRNA you want. If you want to knock down something, this can do it. Also, there are CCK receptors in other cancers like gastric cancer, colon cancer, medullary cancer, and some lung cancers. So we have a team of scientists, and we're hoping we apply to be one of the AACR Dream Team Stand Up to Cancer. And again, to take something like this with our nanoparticles and take it to the clinic, it's going to take a whole team of scientists. I mean, I'm a clinician scientist. We have an oncologist working with me. We have somebody who's a material science engineer who helps design this. I have an imaging engineer, a pharmacologist, a toxicologist, project manager, and we have patient advocates to make sure that we're doing the right things and spending money wisely. So that's taking an idea. You find out there's a problem. There's no way to diagnose pancreatic cancer in the early stage. So we found a receptor and we can target it. So what are the obstacles with translational research today? Well, of course, the biggest obstacle is money. It costs money to take any project from the bench to the bedside. And, you know, is there a lack of funds, misuse of funds, disparity of funds, you know, whatever. It takes a lot of money to develop these ideas. I'm a clinician scientist, and so I can say clinicians don't get protected time to do translational research. They would rather have me in the clinic doing colonoscopies because they think that makes more money and helps more people. But, you know, in the long run, you know, coming up with a cure or discovery for a cancer is, I think, much more important. But it's the getting there that doesn't pay as much as fast. There is this big division between industry, I think, and the NIH and academia. You know, industry likes to come up with their own ideas because they want to have the patent rights and everything. If you come up with an idea, you have to license it to the company and they have to pay you. So they would rather come up with their own. And then, of course, industry always comes up with what I call the Me Too drugs. As soon as one gets a drug, then there's a Me Too drug that comes out and they're all the same, but they just modified it a little bit. So then there's always problems getting patients to go into research studies. And if I can go talk to Congress, I should tell them when they're coming up with the health care bill, they should tell people that if they enroll in a research study, that they'll give them a discount, because that will get more people into research studies and it'll help keep our costs down for health care, because then it'll be covered by the research study. And of course, there's no more one-man bands. You need a whole team of scientists to do a project. So, and the bottom line is does the research really have any clinical relevance? These are a couple of my patients who, with their permission, let me show their pictures. They had failed standard chemotherapy for pancreatic cancer, and they enrolled in some of my research studies. And they're, you know, happy to talk about participating in research. And we need more patient advocates like this that will support research. And then what about our CCK antagonist? What's the potential side effect? Well, it might decrease heartburn. So I think that would be a beneficial side effect. There are CCK receptors, as we talked about, in the stomach. But if we deliver our gastrin siRNA to those receptors, those cells don't make gastrin, so there won't be any harm. But our antagonist, if we block the receptor, we will probably have less acid. These drugs were actually developed 20 years or more ago to treat peptic ulcer disease. And with the advent of the proton pump inhibitors, they have fallen off the wayside. They're no longer under patent. And people have lost interest in them because the proton pump inhibitors are $1 billion business. And so people have forgotten about these drugs, but they might have another use. So you need to think about that when you're doing research, too, is about repurposing old drugs. They may be beneficial for another cause. And I just want to thank some of my collaborators that helped me with some of our research, and, of course, all of my animal volunteers. So I'm happy to take any questions. Yes. That's an excellent question. And there's a couple of possible hypotheses. One is the obesity and the metabolic syndrome, high-fat diets. It's well known that in countries that consume high-fat diets, the incidence of pancreatic cancer is skyrocketing. We know that high fat stimulates CCK. CCK binds to the CCK receptor, and that may stimulate the growth of pancreatic cancer. The other possibility, and although the industry doesn't want to hear me say this, is that for the past 20 years, when the incidence of pancreatic cancer has nearly quadrupled, also has the use of proton pump inhibitors. Proton pump inhibitors raise your gastrin level, and we showed in our study that those precancerous lesions in the pancreas, which everybody has some of those, kind of like people have polyps in their colon, we don't know what triggers them to go on to become carcinoma in situ and cancer. But we showed we could prevent that effect if we used a CCK antagonist, which blocks the gastrin. So it's possible that the proton pump inhibitors that have been overly used and abused in the past 20 years are related to the increased incidence of pancreatic cancer. That's my thought. So, other questions?
Well, in your clinical trials, are you going to be screening the patient tumors to make sure that they have the CCK receptor?
Yes, we are. Yes, we are. So patients will have biopsies before they start in the study, and also about 4 weeks into the study, because we're interested not only in the receptor density and how well they respond based upon their CCK receptor number, and also we want to look at the microenvironment too, to see if our therapy will be increasing the influx of CD8 positive cells and maybe decreasing fibrosis. So we'll be doing a second biopsy in our clinical trial. And that leads to our better understanding of what's going on in humans. I mean, we know it happens in mice, but we want to know if the same thing happens in the humans. Yes? So a transfected cell line with CCK receptors. So all pancreatic cancer cell lines that we have screened and all pancreatic cancers from the operating room that we have screened all have CCK receptors. So we did not have to overexpress it at all in our cancer cells. In the animal that I showed up there that we did the binding, it actually was BXPC3. Which actually doesn't have as many receptors as like PANC1, which is another pancreatic cancer cell line from ATCC that everybody uses. We do, now that we're doing syngeneic models in immune competent mice, we're using some cell lines. They're called MT3, MT5 cell lines. They were derived from organoids from David Tuveson's lab at Cold Spring Harbor, and he took the cells from these pannins and the cancers that developed from the KRAS mutant mouse, and he grew them as organoids and developed cell cultures, and you can put them back into the mice. And they resemble the human cancer very well because they have mutant KRAS, and they also overexpress CCKB receptors, and they make gastrin, which others may not. So they're a good model to use when you're doing immune competent mice. Yes? So the longer term survivors, as far as the people who had the SNP all died rapidly. So if pancreatic cancer, whether you have the SNP or don't have the SNP, is still a poor prognosis. There are some longer term survival patients, and at least in our study, the ones that survived longer had the wild type genotype. The CC genotype rather than the heterozygote AC or the AA, which is the SNP. So it may be related to it, And when we did our study, the statistician, why it's important to have a statistician on your team, he evaluated all the data and made sure that we corrected for stage of disease so that we were sure that the survival was not influenced by somebody lived longer because they were early stage disease. So they corrected for all of those factors. So it may be related to that. Yes. So, okay. Thank you very much.
Okay, in terms of announcements, next week we don't have class. Our next class will be 2 weeks from today. And let's see, file close. And here's Kurt's. Okay, and our next speaker is Kurt Harris. He got his MD degree from the University of Kansas. Then he did a residency at UCLA. He's been at NCI seemingly forever. And he's won numerous awards. I saw him win the AACR Princess Takamatsu Award, where he spoke before thousands of people and gave an excellent talk. And he's published over 500 journal articles. His claim to fame, though, is he's editor-in-chief of the journal Carcinogenesis. So he's going to talk to us today about lung cancer, precision medicine strategy. Kurt.
Okay. This working? It should be. So I think Jill gave you a very nice presentation about pancreatic cancer and how to do clinical trials. And I'm going to be talking about precision medicine. And then I'll use some examples of lung cancer to illustrate the current strategies of precision medicine, including the NCI Precision Cancer Initiative. So, yep. Okay. Nothing's happening. We all need help. Okay. Fantastic. All right. So, when I was going through medical school and learned medicine, this is what I learned. This is traditional medicine, lifestyle, medical history, signs and symptoms. We actually put our hands on patients and did physical exams and various simple laboratory tests and chest x-rays and things like that. So precision medicine is different. It's multi-layered, it's individual-centric, and it's interconnected. And it uses information commons in these various omics, which I'll be talking about. So the idea of precision medicine follows various strategies, such as those called personalized medicine and P4 medicine and so on and so forth. But this was from a NCI, excuse me, it was from a National Academy of Science report in which there was information commons that utilizes various omics that's on the foundation of clinical information and epidemiological information. This leads to information or knowledge network and a better classification of patients. And this for the objective of improving clinical medicine. Whoops. And these observational studies go back to the information commons to improve the process. It also informs biomedical research. The thing that most of us do here is scientists, and there's the informed mechanistic studies also go back to information commons. What wasn't part of the proposal, but actually Vargas and I wrote a extensive review on precision medicine and is prevention research. So let's first describe some clinical and epidemiological data. And as I said, I'm going to be focusing on lung cancer. And this comes from a report of the Surgeon General in 2014 that I and John Sammett and other people contributed to. And this is the 50th anniversary date of the smoking and anti-smoking campaign and starting it. And these are the numbers of individuals who have had premature deaths as calculated by smoking or exposure to secondhand smoke over this 50-year period. It's close to 21 million people. And these are the smoking-related cancers and also cancers from secondhand smoke, and then other causes of death related to smoking. You're probably familiar with this kind of chart in which these are the cancers per 100,000 of the population, year of diagnosis. This goes to about 2014 or so. And male cancer, lung cancer, reached a peak and is rapidly falling. It's about almost not quite half of what it was. And the female cancer, lung cancer, is plateaued off and maybe starting to decrease. So smoking, in the United States, this is the amount of cigarettes per, in terms of smoking as a percentage. And this is obviously the US. It used to be like this, in which over 35% of the population were smokers. There's still populations in the United States in which there's high levels, in the world in which there's high levels of smoking. So lung cancer is increasing in the United States and decreasing in the United States due to anti-smoking efforts, but not in many other parts of the world. Now, not only active smoking, but passive smoking can increase the risk of lung cancer. And this is a watercolor that was actually done by Hirayama, an epidemiologist in Japan. And this was a meeting in 1981, which I and a number of other people attended at Cape Sunion in Greece. And he presented for the first time that wives of Japanese workers who smoked had an increased risk of lung cancer. I will tell you at that time, the vast majority of the people didn't believe him. And, but he proved to be right. And now there's 50, 80 studies convincingly show that being around people who smoke or being exposed to smoke increases your risk of developing a lung cancer. And this is a quotation from Hippocrates. I kind of like these kinds of things, so bear with me. Some men, he wasn't so interested in women in those days, some men and women, I will say, have constitutions that are like wooded mountains, running like springs, others like those with poor soil and little water. Still others like land rich in pastures and marshes, and yet others like the bare, dry earth of the plain. Essentially, people differ from one another in terms of their constitution and risk of disease. So 28 years later, we decided to do a study of lung cancer in never smokers. And I had this idea when I was sitting listening to Hariyama give his talk, and that is there an increased risk of smoking in kids who were never smokers but were exposed to their parents' smoke? And we didn't do this until 28 years later, because in the United States, women didn't start smoking extensively until the Second World War. And so we had to wait until 50, 60 years later that lung cancer occurs. And this is Susan, who did this epidemiological study with us. She was a postdoc at the time. And the conclusions from the study is that parental secondhand smoke exposure during childhood is associated with a dose-dependent increase in lung cancer risk among never smokers in 2 separate cohorts. And it's very important to look at multiple cohorts when you're doing these kinds of studies and others. And I think Jill made that point too. And this was increased risk of lung cancer was especially true with those who had a mannose-binding lectin haplotype with a hyperactive innate immune system. So inflammation and increasing risk. What was surprising to us at the time that these people had lung cancer in their 40s and 50s, and not in the 60s and 70s that most cancer patients are, who are active smokers. So lung cancer is number 1 worldwide in terms of mortality, and our group, and I'm head of a laboratory, that means a department, as you guys know here. I'm studying primarily lung cancer as well as Reed Ryan. Liver cancer is Shin Wang. Pancreatic cancer is Pervez Hussein. And Stefan Oms is studying breast and prostate cancer. So we're studying the major worldwide cancers. And throughout the talk, now I'll be talking about early stage cancer and early stage lung cancer, because that's the opportunity to get the highest cure rates. But there are certain aspects of this that are important in terms of developing risk biomarkers to identify people for priority for low-dose CT, that's computerized tomography, and also early diagnosis, because many of these lesions that are detected by low-dose CT are benign, so you need to know whether it's cancer or not. If there's a diagnosis of cancer and there's a surgical resection, one needs to know whether or not the patient is at high risk for recurrence, and if so, they might benefit from adjuvant therapy, either immunotherapy or chemotherapy. So let's first go to exposure, and the expososome, as it's called. And this was coined by Chris Wild, who's the head of the International Agency for Research on Cancer. And it includes not only the external environment, such as tobacco smoke, infections, and diet, and so on and so forth, but the internal environment that's directly connected, such as obesity, which Jill mentioned, also pancreatic cancer and chronic inflammation. And these together can lead to cancer biomarkers of risk and diagnosis. Cancer genome and epigenome, genetic susceptibility, driver genes, obviously for cancer therapy, but understanding carcinogenesis can also benefit our biomarker studies for cancer prevention and screening strategies. So let's talk about inflammation first. Inflammation can be largely inherited more than acquired. Conditions such as hemochromatosis, or which is a iron overload disease, increases the risk of liver cancer or inflammatory bowel disease, or even pancreatic cancer. So that is associated with familial pancreatitis. But more often it's acquired, and it can be due to viruses, bacteria, and parasitic conditions. And I also like to point out human papillomavirus, because John Schiller and Doug Lowy just received the Lasker Award, which is one of the highest awards in medical science, just quite recently. What isn't an infection are chemical, physical, and metabolic conditions such as acid reflux, Barrett's esophagus, obesity, or smoking in multiple sites. And the global impact in the World Cancer Report that we put together in 2014 is about 2 million cancers per year are related to infection, and other causes of inflammation are associated with many more cancers per year. Smoking, for example, over 6 million cancers per year. Now let's go further into inflammation, and these are a series of studies that we have done and published on lung cancer, including looking at health disparity, comparing Afro-Americans to European Americans. And the first was a prospective study that involved about 100,000 people, and we found that in a nested case control study that IL-8 and CRP, that's in European Americans, increased the risk of lung cancer. At time of diagnosis, there was also continued increase in IL-8 and IL-6, but now we had enough cases to look at both Afro-Americans and European Americans. And the Afro-Americans had, other than these two, they had increased in interleukin-1 beta and IL-10, which is an anti-inflammatory cytokine. And poor survival. The inflammation also is related to survival in case of lung cancer patients. And this is again controlled for stage and the various other things that are important. So again, IL-6 and IL-8 increase the risk of dying, and nanobinding lectin, which I talked about a moment ago, increased TNF-alpha in European Americans, and again, IL-10 and IL-12 in this case in Afro-Americans. So I mentioned IL-6 and IL-8, and this is the earliest stage of lung cancer, that's stage 1A. So it's less than 3 centimeters, no evidence of metastases. We look at the nodes, and when both of these are elevated in this Kaplan-Meier plot, so this is 100% survival and 0% survival, and this is survival in years. One can see that if both are low, relatively good survival for this early stage cancer. If one is high, intermediate, and if both are high, 75% of the people are dead, even though they had the earliest stage of cancer. I'll get back to that in a moment. All right, how about genome? A number of years ago, quite a number of years ago, actually, several hundred years ago, it was recognized that chimney sweeps had an increased risk of scrotal cancer, and this was due primarily to various polycyclic and aromatic hydrocarbons such as benzpyrene, aflatoxin, that is a fungus that grows on moldy corn and grain, also produces a very potent carcinogen called aflatoxin, causes liver cancer, and I mentioned cigarette smoke, which has multiple, in fact, it has about 60 different known chemical carcinogens. So it's a real witch's brew of carcinogens. and no wonder it causes so much cancer of multiple sites. So quite a number of years ago, the first kind of question that was asked by our group and followed by others was whether or not these specific carcinogens, such as aflatoxin, benzpyrene, ultraviolet light, or more recently, this renal toxin and carcinogen, whether it caused mutations in the p53 tumor suppressor gene. And the answer was yes, and it had caused very characteristic mutations associated not only with cancer, but associated with the mechanism of action of these various agents. Now, more recently, Sir Michael Staton and his colleagues at the Sanger Institute in London have utilized exome sequencing and trinucleotide analysis of the signatures, and have found that this is a signature for tobacco, this is a signature for ultraviolet light, and so on and so forth. So there are about 30 different signatures. About half of those are associated with specific exposures, and a few are associated with just aging. And people get older and they don't have these other risk factors, they still get cancer, and they have a characteristic type of mutation signature. Now, lung cancer, a number of years ago, it was just classified as small cell adenocarcinoma or squamous cell carcinoma. And then in '87, KRAS was discovered. And in 2004, EGFR was discovered. And lastly, in 2014, this is the fairly up-to-date, a number of different types of mutations and fusions, such as ALK fusions, were discovered and added to the genomic landscape of lung cancer. I will say that most of these that have been recently discovered are in never smokers that come into our clinic. And about 14% of the lung cancer cases that come into clinics now are never smokers. Now, is it passive smoking or something else? So the majority of cancers that we still don't have therapy for have KRAS mutations, or they have p53 mutations. So the p53 gene is the most commonly mutated gene in human cancer, including lung cancer, particularly squamous cell carcinoma, but also adenocarcinoma. And as a wild type gene and its protein, it's a tumor suppressor, but there are multiple mutant types. And some of these have lost function as a tumor suppressor and varying degrees of loss of function, and others gain of function as an oncogene. So the p53 gene can switch from being a tumor suppressor gene to a tumor oncogene. And this was confusing in the initial 10 years that p53 was being investigated, but it's quite clear now. And in fact, those that have a gain of function mutation here in red, again, this is 100% survival, this is 0% survival, this is time in months, so this is 5 years. So these individuals have a poor prognosis, controlled for various other things than those who have just a loss of function. So let's look at the transcriptome. So first, non-coding RNAs, such as microRNAs. And these non-coding RNAs were first discovered by Victor Ambrose in C. elegans. And that was in 1993, I believe. And in year 2000, his colleague, Gary Repkin, found in human cells that there were these small microRNAs. And they function by inhibiting hundreds of genes, the protein output of hundreds of genes that they repress by either destabilizing the RNA or, to a lesser extent, inhibiting translation. It wasn't until 2004 that Carlo Croce discovered that microRNAs are discovered in human cancers, particularly chronic lymphocytic leukemia initially. And it's been now shown that microRNAs can predict risk, diagnosis, prognosis, and therapeutic outcome. So most of you, I'm sure, know about microRNAs. They function largely by translational repression. So this is the microRNA, so the protein isn't made, or by, and this is actually more frequent, by binding to the message, frequently has a nucleotide that's different than what the binding microRNA would recognize, and the message is cleaved. But microRNAs can also bind to proteins that alter differentiation, and also microRNAs such as miR-21 and miR-25 can bind to total receptors, both in human and mouse, and lead an increase in IL-6, which I talked about a moment ago, and TNF-alpha. So we started a number of years ago, actually in 2004, 2005. This is one of my former postdocs, who actually is associate professor now. And the hypothesis was that microRNAs associated with lung cancer diagnosis and prognosis. And we found that the microRNA profiles were significantly different between primary cancer and those of non-cancerous tissue, and among different histological types. So you could recognize squamous versus adeno versus small cell. And we found there was an increase in miR-21 and 155 and 106, and a decrease in lead-7 that were associated with diagnosis and prognosis, again, in the earliest stage of lung cancer, stage 1. This led to a collaboration in which we participated in looking at multiple cancer types. In fact, the 8 most common kinds of cancer at that point. And there were, I think, about 300 microRNAs identified. When we did this study, there are well over 2,000 now that have been identified. But it was a simple kind of study. We just took multiple cancer types and asked the question, which of these microRNAs were most highly expressed? And we found that miR-21 was expressed in 6 of these major cancer types. So miR-21 is upregulated now in over 18 major cancers, is a biomarker of poor prognosis in 14. So this is another fairly early study that we did. These are my Japanese postdocs at that time. And we compared a Maryland cohort from the Maryland, Baltimore area to a Norwegian cohort versus a Japanese cohort. And these are Kaplan-Meier plots again, 100% survival, 0%. And this is high miR-21 expression in the tumor versus low, this is median, above the median or below the median. Now you can look at this and say, well, maybe the Japanese are better surgeons than the US surgeons. But importantly, this is looking at relapse-free survival. And here we're looking at cancer-specific mortality. And I always prefer cancer-specific mortality than relapse-free. So this is observation is true not only in lung cancer, but in multiple other kinds of cancers, as I mentioned. And these are just some examples of various studies, and all of these have been confirmed by 2 or 3 additional studies. So miR-21 is an indicator of poor prognosis in multiple cancers. So what's the mechanistic underpinning of this microRNA in human cancer? First of all, it's in an area of amplification. And in the latest report of lung cancer and various kinds of genetic changes, gene amplification at this site is now a important contributor to diagnosis and prognosis. And we think this is miR-21. Decreased transcriptional silencing, talked about that. We showed in a PNAS paper a number of years ago that EGFR mutations led to an increase in miR-21. tumors that have EGFR mutations of lung cancer don't have KRAS mutations and vice versa, but KRAS also increases in MIR21. And various inflammatory agents, as David Baltimore shown, increases STAT3, which is a transcription factor for MIR21, and various types of DNA damage also. So what's downstream of MIR21? Well, it inhibits a whole variety, and this is just a snapshot of some of these targets that are important in tumor growth and focal adhesion, anchorage, independent growth, migration, and also affects certain DNA repair genes. And as I mentioned earlier, that it leads to activation of toll receptors and inflammation. And some of these studies were initiated again by the Crochy lab, in which they found that cancer cells release in exosomes, these are small pockets of membrane-bound vesicles, less than 100 nanometer, that carry these microRNAs to immune cells to activate the toll receptor, or they also carry them to myelocytes, muscle cells. And one of the most devastating associations with human cancer is cachexia. Cachexia means that they have wasting of all their muscles and fat. So this is a condition which there's no good therapy of, and these individuals, over a period of just a few months, they look like they're starving to death, and that's the and they die, and so that's cachexia. So MIR21 and through again the inflammation may be playing an important role. That's being tested in the clinic right now. All right, so I talked about microRNA as a transcriptome. How about protein coding genes? So we did something called random forest. That's a fairly sophisticated way of doing analysis, in which we identified 4 genes that are associated with poor prognosis. The first is increased NXPO, which is involved in transport from the nucleus to the cytoplasm of microRNAs and other things. BRCA1, which you relate to breast cancer, but high levels of BRCA1 actually are genotoxic and cause instability. And HIF1 alpha associated with decreased oxygen tension, and a tumor suppressor gene discovered here at the NCI, DLC1. And this is 12 different independent cohorts, again, stage 1A cancer. And as one can see, if you look at the lowest quartile, which is the green, the middle tertile, or the highest tertile in each of these cases leads to poor survival. So again, this is a principle of looking at multiple cohorts when you have an interesting finding. And if you find it in multiple cohorts, it probably is valid. This is just comparing stage 1A versus 1B. So I mentioned stage 1A is 3 centimeters, stage 1B is 5 centimeters, and in both cases there's no evidence of metastases in the lymph nodes, but I'll get to the point that they do have micrometastases. So this is looking at stage 1. These are the multiple studies. Most of them just looked at overall survival, which is less accurate than relapse-free survival or cancer-specific survival that we prefer, but in each case is associated with poor prognosis. So next kind of question we ask, and you can ask, is how about if you combine biomarkers? And the principle is if they're statistically and mechanistically independent of each other, it makes sense to do that combination. And so this was a hypothesis that Hiro and Cherry tested and comparing the 4 protein coding genes with microRNA miR21. So this is a Japanese study, and this is again relapse-free. So this is comparing the 4 gene signature that I mentioned before with, which is up here, with miR21, which is shown here. Each of them leads to associated with poor prognosis, and you put the 2 together, again, it's a significantly decrease in prognosis. And this is the US-Norway cohorts, and this is 4 gene signature, poor prognosis. This is miR21 for prognosis, and again, if you put them together, much poor prognosis. Again, this is cancer-specific mortality instead of relapse-free survival. So what's the principle? Well, the principle is if you take non-coding RNAs and coding RNAs and you combine them, you should get low risk, intermediate risk, and high risk. Why is this might be the case? Well, in both the RNAs, they may give you agreement, but each biomarker gives you some false positives, but they're different from each other. So these are misclassifications compared to better and more specific survival. Is this true only in lung cancer? The answer is no. We and others have found that it's true in colon cancer, esophageal cancer, adenocarcinoma I just mentioned, and also in breast cancer. So now let's move to the epigenome. And this is Anna Robles, who's in the lab, and is a staff scientist, and she tested the hypothesis that an integrated biomarker classified of stage 1 lung cancer, adenocarcinoma, based on independent mRNA, microRNA, and DNA methylation biomarkers, this is the epigenome biomarker, will further improve prognostic classification. So this is putting 3 biomarkers together. Now, it had already been published by Manuel Estela that these 3, 4, 5 genes that were DNA methylated were associated with poor prognosis. You said, well, why don't you look at all 5 of them? Well, this is by univariate analysis, but if you did multivariate analysis, which we did, we found that they're all dependent on each other. So we chose HOXA9 to do the analysis of, which is very representative of the total. In fact, maybe the most representative. And we added it to the miR21, the 4 gene signature, HOXA9, miR21, the 4 gene signature is here. And we found that if all 4, actually all 3, are positive, that's in the red, the prognosis in stage 1 lung cancer, in stage 1A lung cancer, in stage 1B, again, these are 5 centimeter in size, these are 3 centimeter size, this is both, are associated with poor prognosis. So these are statistically and mechanistically independent of each other, and you could show this by doing these kinds of analyses in multivariate analysis. So now let's look to the metabolome. That's looking at metabolism, tumor metabolism. And we've been interested in this for a few years now. And Maya is now at the FDA, and Evie is now at Ohio State. And our initial study was an unbiased metabolomics, and that the hypothesis will discover biomarkers associated with risk, diagnosis, prognosis, and therapeutic outcome, again, of lung cancer. Now, one of the liquid biopsies, one of the fluids that we decided to use was actually urine. And here's a nice painting of a Dutch physician looking at urine. And it's been reported that Hippocrates and physicians at his time in history tasted urine for diagnosis of disease in his patients, such as diabetes. Others didn't know what diabetes was. When I went to medical school many years ago, as Terry mentioned, we didn't have to taste urine, thank God. But we had some very simple tests for doing that. So this is another very short quotation from Armstrong, who's this physiologist. From the liquid window through which physicians felt they could view the body's inner workings, urine led to the beginnings of laboratory medicine. So Evie actually did an unbiased screen of literally thousands of potential metabolites, and by, again, random forest, came up with 4 of them that were statistically and mechanistically independent of each other. Creatine riboside, which had never been described before, and actually the NIH is going to have a patent on that as a metabolite. As I said, never been described before. You can't patent proteins that are already known or molecules that are already known, but something that's never, ever, ever, ever been discovered, you can still patent. And N-acetyl neurominic acid and cortisol sulfate, and this is the glycolipid that we've yet to discover. So these are Kaplan-Meier plots. If you put all 4 of these together, again, this is lung cancer. There's a very poor prognosis, less than 10% at 5 years, if all 4 of these are positive in the urine. And we can now measure these in serum and plasma. So this is just the hazard ratios that go up when you make these kinds of combinations. Now, one of the important questions when we first started doing these studies with, say, creatine riboside was it found in tumors, and was what you found in tumors related to what you find in urine? So yes, it is found in lung cancers at relatively high levels compared to the non-tumor area. And if you compare creatine riboside in the tumor versus that which is in the urine, this is the correlation analysis, MAR about 0.6 and a 0.006 significant factor. And right now we're asking the question if serum or plasma correlate with this and triangulate and give us another set of biospecimens to do these kinds of analysis. With our colleagues at Vanderbilt, who have done a prospective study of Afro-Americans and European Americans, again, about 100,000 folks. And so we did a nested case control study of lung cancer in these individuals, and we found that in the multivariate analysis that the odds ratio are quite high with the creatine riboside and the N-acetyl neurominic acid. That's actually gets the attention of the diagnostic community. However, when we looked at Afro-Americans, it was not elevated. So there is a health disparity here, and that's something we're quite interested in finding out about. So how about using this as a screening for lung cancer in high-risk European Americans? If you put these together along with known risk factors such as smoking and other risk factors, you find that in this ROC curve, which looks at sensitivity and specificity, this is a straight line, but the combination is in green, and the odds, the association under the curve is actually it's over here. It's close to 0.95 between 8.6 and 9 if you put everything together, which is here. And this is something that can be clinically useful. And It's something, again, the NIH has patent rights on. As you all know, investigators at the NIH get very, very little money from patents. But the patents are, and the research outputs comes from taxpayers' dollars. So I mentioned low-dose CT a moment ago. And this is a landmark study that was published in New England Journal of Medicine in 2011, in which they compared chest x-rays with low-dose CT. And chest x-rays were more or less standard practice at that time, but we knew that they weren't very useful, only detected later stage of cancer. But in this study, they found that low-dose CT actually reduced cancer mortality by 20%. And this is some of the specifics back here. About over 50,000 individuals were analyzed. These were primarily heavy smokers of this age group. There are two different groups, about the same number of individuals. There's a very important finding, though, and that's the false positive rate is about 96%. And I'll make that point a little more stringently in a moment. The kinds of cancers, lung cancers, that were detected by low-dose CT were primarily stage 1A and 1B lung cancers. So about 60% of these lung cancers were the very smallest ones, the ones that we have the greatest opportunity for curing. Now, I mentioned the high false positive rate. So this is an illustration of what this means. So this is a stage, and these are all chairs in the audience. Michael Wolftrap went to hear Sting a couple nights ago, 3 nights ago actually. Fantastic, I might say. And low-dose CT detects 3, maybe 3.5 of these individuals screened. And these are the people who were screened. So this is a problem, because if you get false positives, that means people may undergo surgery that don't need to go undergo surgery, or be told they have cancer and they don't really have cancer. So low-dose CT is not only standard practice now, but it's being paid for by Medicare, and hopefully this will continue, the Affordable Care Act. So these are the current risk factors for being eligible. This is the age grade. This is a 30-year history of smoking, quit within 15 years of current smoking. But we need to identify high-risk individuals outside this criteria, including what I mentioned, about 14% of people coming in the clinic now are never smokers. They would never have funding for these kinds of assays. Second is prioritization. And maybe I'll go to about to the end. Next slide for that. But there's now about 10 million people that are eligible with these criteria. So there's a need to prioritize those individuals for low-dose CT, because the current capacity is less than 200,000 per year in the United States. And we're talking about 9, 10 million people who should be screened. And many of those people screened are negative or have false positive rates. So it would be important to identify individuals for priority screening, partly for cost reasons and access to care. Second is, if a lesion is found by low-dose CT, and 96 of the 100 lesions are benign, which ones are actually benign? So we need better diagnostic biomarkers to make the clinical diagnosis. Today, about half of the people going to surgery in the United States, in at least many different hospitals, that have a positive lesion by low-dose CT, have a benign tumor. So doing surgery and cracking, as the surgeons say, cracking someone's chest is not a benign procedure. And so you would like to minimize those people who you do surgery on and you remove a benign tumor instead of cancer. And then there's the question of prognostic biomarkers, even for stage 1 lung cancer. So the goal of precision medicine is to identify, or one of the goals is to identify 20% of the stage 1 lung cancers whose cancer will reoccur. So pathologist doesn't find anything in the nose, but about 25 to 30% of those people have micrometastases. And one of the questions is, if you get therapy for micrometastases, is that going to be more effective than if you wait till you have a recurrence that's the size of a golf ball? That's a hypothesis to be tested. So again, we have stage 1A, stage 1B. These are the different sizes. You might both go undergo surgery, but some people are going to be low risk of developing metastases, either stage 1A or stage 1B. But some of these people are going to be high risk. So one of the goals of precision medicine is to use these various omics, coupled with good clinical information and epidemiological data, to decrease the false positive rate, decrease financial costs, improve patient care, and guide mechanistic studies. And part of the patient care, those are low risk of recurrence, observation, and maybe doing low-dose CT at varying time intervals, but those at high risk, giving them the option of adjuvant immunotherapy or chemotherapy. So these are the colleagues who have done most of this work. Many of them have their own laboratories now. So I want to thank them, and I'd be happy to take any questions that you might have. Thank you. So how many of you have a member of your family that has lung cancer? I'm surprised no one's holding up their hand. Because I have, and probably some of them. Not me, fortunately, but so it's a very common cancer. In fact, it's the most common cancer worldwide. So questions? Yes, ma'am.
For some people with small coli,
Yes.
they live long life
Yes.
without cancer, including obviously lung cancer. Anyone study those people with small coli without that cancer?
So the question is a good one, and it's that there are smokers who never get lung cancer. They might die of chronic obstructive pulmonary disease. They might die of emphysema. They might die because they cause a fire. There's lots of where they have a heart attack. But there are people who are smokers into the 70s and 80s, and maybe even 90s. But those are fairly rare. But those are very interesting cases to investigate. And just like pancreatic cancer that Jill was talking about, those people who live 4 or 5 years, what's different about them than those that do not? So looking at extreme survivors is one of the priorities at NCI now, to try to identify those individuals and why. That's the important question, why? What's the mechanism of long-term survival? Is it more than just by chance? Other questions? Yes. Yes. So the question relates to health disparity, particularly Afro-Americans versus European Americans. And there are some studies in which economic status and a whole variety of other things, smoking menthol cigarettes versus non-menthol cigarettes, are actually part of the study. And the risk factor study that we did with the people at Vanderbilt, they did all of that. In about half of the cases, individuals that were that participated in the study were Afro-Americans versus European Americans. So one could, once you control for that, show that, in fact, many of those social factors that you're talking about, or may be talking about, are not the reason for survival or these various risk factors. In fact, Reed Ryan in our group right now has some evidence that Afro-Americans may actually live longer and have less cancer, lung cancer, than European Americans. That's a very startling finding that has been shown in a couple of studies now. So why? And so what are people doing? They're looking at SNPs to be associated with different cancer types and risk. And Jill talked about that. And that's a very active area, looking at gene expression analysis and various other things. So health disparities, again, it's a high priority for the National Cancer Institute, not only CCR, but also Division of Cancer Epidemiology. And there are some people who are very much involved with that, and I hope you're one of them. No, increased. No. That was a very important question. What's the criteria for access to care and diagnosis? And in the study that I talked about, in some of these other studies, that has been addressed. So it's not just access to care, or it's also in terms of Afro-Americans versus European Americans, even though in average, Afro-Americans have less access to care. But that's not the reason for the lung cancer risk.
What about the type of cigarette?
All right, so yes, for many years, menthol cigarettes, which are 80% of the cigarettes that Afro-Americans smoke, compared to about 20% of European Americans, that was thought to be a cause and a contributor to lung cancer in Afro-Americans. And the rationale was, well, menthol sort of anesthetizes the throat, and so you can smoke and inhale more deeply and hold the smoke in more. And so our collaborator, Bill Block, who used to be here at the NCI, published the definitive paper about 2 years ago in the Journal of the National Cancer Institute that that menthol cigarettes were not responsible for, they weren't any different than regular cigarettes in Afro-Americans or European Americans. And again, Reed Ryan in our group has found a similar thing in the cohorts that we were studying. So very nice hypothesis, mechanistically very attractive, but happens to be wrong. And that's called the scientific method. And this is maybe one of the last things I'll say. It's a not more question. Frequently, I ask when I give talks, and the ones I seldom refuse are those by graduate students and postdocs at various medical schools, and they usually have one person they can invite each year. So the question is, how many of you are trying to prove your hypothesis? And I'm not going to ask you to hold up your hand or not, but about half of the people who are graduate students and postdocs in the United States in these multiple schools that I go to say they're trying to prove the hypothesis. That's not the scientific method. You're trying to disprove the hypothesis. And trying to prove the hypothesis that your boss may have leads to manipulation of data and If the mentor isn't actually looking at primary data, can lead to retractions of paper, including the very best scientists you know, including those that are producing papers in Cell, Cancer Cell, in lots of Nature. A lot of those papers are being retracted because the postdoc or graduate student is trying to prove a hypothesis that is in favor by the mentor. So don't do that.